We're going to get started on time today. Hello. We hope you have had a great break networking, and the sponsor for that breakout session was Leadient Biosciences. We hope that you were able to get some quality time in there. Thank you for joining us here for track 4, What's New, Innovative in Rare Disease. And this is the last track of the afternoon. I'm Alan Gilstrap. I work for Alcuos, a startup gene therapy company. So this information will be very interesting to me as well. And I'm your host, room host for this last breakout session. What is druggable? New therapeutic targets in rare disease. As a reminder, this track is being live streamed, and we would like to welcome our remote attendees and remind everybody that we have the remote app. So if you have a question that comes to your mind during the session, you can put the question on the app and we will try to get to all of the questions at the end of the session. We will also have a mic for you in the room, so if you're brave enough to stand and ask your question, I'll be there to hand you the mic. As all of us know, today only a small amount of rare diseases have approved therapies, but with the advancements that are being made and the increased knowledge that is being gained in rare disease, we have a better potential for the development of therapies. To help us explore this topic and to share with us their experience, we have Jason Sicklick, Associate Professor of Surgery at UC San Diego, Moore Cancer Center, Division of Surgical Oncology, and Yi Liu, Director of Gene Transfer Technologies at Regenix Bio. Let's welcome them. Thank you. And I'll leave it to you.
Fantastic. Thank you very much. Good afternoon. Thanks, everybody, for coming, and welcome to San Diego. So today I'm going to talk about what's druggable and new targets, and I'm going to focus this on cancer, but use cancer as a paradigm, not necessarily as the end-all be-all of this. So here's my disclosures. So when we think about cancer, we think about it based upon histologies. We talk about bladder cancer, breast cancer, lung cancer. And this has truly become a religion, if you will. This is the way pathologists report it. This is the way surgeons talk about it. This is the way medical oncologists talk about it. And we divide up the way our disease teams are based upon histologies. And this obviously translates into how clinical trial designs are made. So when we think about developing a clinical trial, we take a patient group with a given disease, and essentially one of 3 things can happen when we treat those patients. Their disease can get better, it can get worse, or it stays the same. But one of the things that, as we're learning, and now we're in 2019, we understand a lot more about biology of tumors. And so one of the things we need to start thinking about is a different paradigm of studying cancer biology and treating tumors. And that's thinking about the biology of the cancer itself. So cancer is a disease of the genome, and in the last session you heard a lot about genetic sequencing. And every day we're exposed to things that can cause cancer: carcinogens, viruses or infections, radiation, as well as just mistakes in DNA repair. Every day that we live, there's cell turnover in our bodies and potentially typos that can occur in our cells, DNA replication. And all it takes is one of those typos to then lead to potentially developing a cancer. So when a gene gets altered, that gene, that genetic code, gets transferred into a protein, and those proteins then conglomerate together, if you will, into a signaling pathway where basically one activates the next, and ultimately this leads to cancer. So I said to you before, we talked about the promise of precision medicine here. And if the promise of the precision medicine would be to take that genetic information and be able to start thinking about how we can individually target each patient so that we can think about giving the right drug to the right patient based upon some of their genetics. And so really this is a matter of sequencing these patients, we then stratify them, and then we try to match a therapy to them. So this is really a shift from where we've been in the past. All of the original chemotherapy agents were really essentially poisons, if you will. And they weren't made to specifically target any one histology or any one tumor type. They were basically developed or identified as poisons that essentially killed all cells, and that's why patients lose their hair, because our hair is rapidly dividing cells, have nausea and vomiting and diarrhea, our GI tract's turning over about every 4 days. And so all these different rapidly dividing cells are also targets. So could we think about targeting tumors or targeting pathways with a single kind of targeted agent? So the traditional approach for precision medicine that's evolved over the last several years is really identifying patients and saying, we've got, as an example here, 3 patients. Patients 1, 2, and 3. Patient 1 has markers A, B, and C, 2 has A, E, and F, and then 3 has A, B, and F. And then we say, let's find a commonality between all these 3 patients, and we're going to give them a drug. So that's going to be drug A. And so this has evolved into what we call either one of 2 things, either basket trials or umbrella trials. Basket trials essentially saying we're going to take all patients that have given gene mutation X, whether it's lung cancer, colon cancer, liver cancer, you name it, and we're going to give them that drug. Versus the alternative being is what we call an umbrella trial, where we basically say we've got this group of drugs that we have available, and we're going to identify patients with lung cancer that can get A, B, or C, or A, E, or F, or whatever that combination may be. So there's lots of studies going on around the world. So I sort of pointed out some of the larger key studies that have been currently underway or have completed over the last several years. And you can see here that there's everything from the United States, obviously, to Israel, France, Australia, as well as the UK. And all of these trials are basically treating patients in a histology agnostic fashion, meaning basically doing sequencing and then treating these patients based upon that. So what's the early evidence for this? So the first trial that was done was reported in 2010. And basically the point of this trial was to say, we're going to take patients independent of what their disease is, and we're going to do some molecular testing. And this is early on in the days of molecular testing. We're going to get a biopsy on them. We're going to do some tests on their tumor. And we're going to figure out if we can find a match for these patients. If we can, we're going to give them that drug. And if we can't, they're just going to get whatever standard drug they get, or dealer's choice, if you will. And what this study did was they looked at what we call the progression-free survival, meaning the time that it takes for your tumor to grow on the new line of therapy, as shown in blue here, as compared to the prior line of therapy that they got. And what you would expect is, in general, as a disease progresses, in its evolution, the progression-free survival, or the time period that it takes for the tumor to grow, should get shorter. But in the scenario here, what they basically said was, we're going to look at the patients where there was a greater than 30% prolongation of that time period with treatment. And it turned out a quarter of patients that got molecularly matched therapy, the blue bar was greater than the yellow bar, and basically it was greater than 30%. So the next big study to come out was about 2 years later, and this study basically said, we're going to take the patients and we're going to do a similar approach, and we're going to use drugs that are available in our clinical trials in our repertoire. And this study was done at MD Anderson. It was called the Predict study. And basically said, we're going to match one drug to these patients. And what you can see here, this is what we call a waterfall plot, where basically anything that's on the positive axis is basically tumor growth. Anything that's on the negative axis is tumor shrinkage. And anything that's between these 2 black lines here is what we consider stable disease, meaning that the tumors either are shrinking or growing, but they don't meet the criteria for what we'd consider an actual response based upon measurements. And what you can see here is if patients got some kind of match therapy, meaning that they had their target, they had a drug, match them together, the response rates were about 50%. Versus if patients just got unmatched therapy, meaning they just pulled whatever drug off the shelf, or they got put on a phase 1 trial of some sort, that didn't molecularly match their targets and their tumors, response rates were about 15%. So subsequently, some of my collaborators at UC San Diego went on to do a large study of about 85,000 patients where they basically pooled data from a bunch of different studies. And what did this basically show is that if patients with cancers are getting non-precision targeted therapies, meaning that you're giving them a drug that's supposed to act on a given target, but they don't have the target, then response rates are somewhere between about 4 to 7%. If you give them cytotoxic chemotherapy, meaning giving them some poison, response rates somewhere in the order of about 12 to 16%. If you give that patient a targeted drug against the target that they have, response rates are about 30%. So you can see here we're sort of making this iterative steps forward in terms of how we can start doing a better job at treating patients. But one of the challenges to this approach is two things. One is what we call a malignant snowflake, meaning that every tumor is different. No two tumors are the same. If you will, every single tumor is rare in and of itself, because if you take two, even breast cancers, they might look the same under the microscope, but genetically, they're often very different. And that's often because of what we call co-genetic alterations, meaning that patients might have more than one alteration in their tumor. And so this was a study that our group did, and I'm not going to get into the sort of details of this, but basically what it turns out is there's high percentages of co-occurring mutations in patients, meaning that if you go back to that original picture I showed you, you had patient 1, 2, and 3. Patient 1 had A, B, and C, 2 had A, E, and F, and 3 had A, B, and F. But we all just decided arbitrarily that patient was going to get drug A, or all three of those patients were going to get drug A. But that is precision targeted, but it's not necessarily personalized to that patient. So another way to think about this is this. If we've got a table with one leg, if you take out that leg, table falls. If you got a table with 5 legs and you take out one of those legs, the table is probably not going to fall. It's no different than thinking about if a patient has 5 different mutations and you target one of those mutations, there's 4 other pathways in these tumors that are driving this. It's no different when you think about other diseases in terms of starting to think about a shift in the paradigm of how we think about treating diseases. And so you can imagine if you can start targeting more of the legs, taking out more of the legs, that table is more likely to fall. So we can start thinking about how to personalize therapy and really truly individualize it for the patient. And that's really a thinking about using combinations of drugs, even thinking about altering the doses of drugs, because we're all different. There's no reason why I potentially should get the same dose of a drug that somebody who's half my size should get. We all have different metabolism, we all have different sizes, we're all unique, we're all sort of our own individual rare. So can we combine this together and think about like a personalized and precision approach? Precision in that we're targeting an individual target and personalizing it for each person. So everybody becomes an N of 1, okay? And so in the situation, could we think about changing the paradigm and saying patient A or patient 1 is going to get drugs against A, B, and C, and patient 2 is going to get drugs against A, E, and F. So this leads us to the iPredict study that we developed here in San Diego. This was developed by myself and Dr. Rizal Kurzrock, who's head of medical oncology at UC San Diego. And we basically came up with the idea of, could we start thinking about treating patients with incurable diseases, with specifically focusing on diseases often where there's no good treatments, which is essentially is a lot of the rarer types of cancers, and taking them and doing some kind of customized combination therapy. This was pretty ahead of its time at the time. It took us about 1.5 years to get this through our institutional review board, but in 2015, we were able to get this IRB approved and get the program activated. Accrual was slow to start off with, and now as of just about 2 weeks ago, we've now enrolled almost 500 patients. We've treated about half of these patients, and we've molecularly matched about 40% of all the patients and 82% of those that we treated. And so how did we do this? Well, you heard about sequencing in the last talk. We didn't do whole exome or whole genome sequencing. just because at the time there wasn't a clinical grade version that we were going to use. Working with a company called Foundation Medicine, which is based out of Cambridge in Massachusetts, we basically worked to combine doing tumor DNA sequencing, and then as the technology evolved, we started adding on more tests to this, and so that such that the protocol was really made to be flexible, and we need to think about, as we're designing clinical trials, including flexibility in that. A trial design that might have taken, it took us 1.5 years to get that trial written, approved, and open. Things change, and science is changing quickly, drugs are changing quickly, and tests are changing quickly. And so as different parts of the test became available, we added those on. What did we do? We took diseases that we said, number one, are going to have a high risk of mortality, and see if we can change these around, change the biology of these with these treatments. And so a lot of these are actually rare diseases, so things like gallbladder cancer, liver cancer, bile duct cancer. These are all rare diseases that we started off, and those were our sort of principal targets. And so the way the trial was designed was we basically said, we're going to take patients that have had at least one therapy, that had failed it, and they have to have either metastatic or unresectable, meaning unremovable disease. We're going to consent them. We're going to send their tumors off for sequencing. If there are no mutations present in their tumor, then it's up to the treating physician. They can give them whatever they think is the most appropriate. But if they have a mutation present, we're going to discuss them at our molecular tumor board, which is a evolving concept in medicine, where basically, as I said to you before, we might have our tumor boards where we say, okay, at UC San Diego, every Tuesday at lunchtime, we have our GI tumor board where we talk about GI cancers, and Monday mornings is breast cancer conference. We're not going to talk about histologies. We're going to talk about the molecular biology of a given person's tumor. And so we talked about each patient's tumor, and we've developed a regimen based around that. And so in the first cohort of patients that we reported in Nature Medicine about 6 months ago now, we looked at the first 149 patients. And of those, we wound up treating 83 of these patients, and we wound up having 66 patients that were inevaluable. And I'll get back to that. Of the patients that were treated, average age was about 62 years old. This included a significant number of these hepatobiliary and pancreatic, these rare types of GI malignancies, as well as you can see here, brain tumors, you can see here head and neck tumors, and then others was a smattering of multiple other rare types of tumors, including sarcomas. We looked at the mutations in these tumors, and everybody had some kind of mutation in their tumor. So there wasn't a single patient that had no mutations. And it turned out that patients had a median of about 5 alterations per tumor, meaning that there was a pretty significant number of alterations in these tumors. So not just one, as we would expect a targeted agent to go after. Now, in order to better understand, and sort of if we go back and think about that table analogy for a minute there, we said we're going to have a matching score. How many legs are on the table, and how many legs did we target? Or in other words, if we looked at the number of total alterations, how many of those were targeted? And that gave us a matching score. And so based upon the matching scores, we would then say we've got a patient with a mutation in BRCA2, for instance, and that can theoretically be targeted with a drug called cisplatin. And this patient might also have these other pathway targets. Well, in the scenario of the first scenario here, if the patient got this drug, we would actually consider this a match for the BRCA, and this would actually turn out to be the standard of care combination of drugs that are FDA approved for gallbladder cancer. Now, if we gave this patient a targeted regimen, they would potentially get a drug called everolimus, which would target all 3 of the alterations. So we would say, okay, this is a 1 out of 3 or a 33% match versus a 100% match. If we looked at the patients that we matched, a majority of these patients are treated, the majority of these patients were matched. There was a small fraction of these patients that were not matched. And then we dichotomized them and said, how many patients had high matching scores, meaning more than half of their alterations were matched versus fewer than half. And that basically brings us down to the sort of box down here, where basically 28 patients had high matching scores and 55 patients had low matching scores. And if you go by some of these the trials that I was showing you before, they might test for 10 genes. But it turns out there's a lot of heterogeneity in all these tumors. And so everybody's tumors is a little bit different. And so I'm going to give you a couple examples of how we've used this approach. And so first patient I'm going to talk to you present as a patient that has a gastrointestinal stromal tumor, or GIST. This is a rare type of tumor that occurs in the GI tract. There's somewhere about 5,000 cases per year in the United States. These tumors are most often driven by another gene called KIT, but there's a subset of these that are driven by this gene called BRAF. And it turns out several years ago, there was a treatment developed for targeting BRAF, and it turned out this patient of mine, was the original case report of taking a drug that targeted BRAF and giving him this drug. And lo and behold, this was a case report, and the patient was actually being treated at MD Anderson at the time. You can see shrinkage of that tumor. So, theoretically, the match worked. But as you can see there, though, it didn't completely shrink the tumor. It didn't go away. So fast forward several years later, he's now on his 9th line of drugs. He comes down to San Diego to see us to get another opinion at our GIST program that we have. And we see him, and it turns out he's got lung metastases, so spread to his chest. He's got disease down in his pelvis here. And so in the meantime, he's been added on to another drug, basically that targets downstream of this original target that had been gone after. And he's progressing. So we do additional sequencing, and it turns out he has an additional mutation in a gene called CDKN2A, which basically regulates the cell cycle, meaning cell division. So in this situation, here we are, he's got this BRAF mutation, it's getting targeted, but this is not being targeted at all. And so in that scenario, we gave him a drug, and I lost the slide. In that scenario, we gave him a drug to target that, and it actually shrunk his tumor down enough that we were able to take him back to the operating room and clear out all of his disease and remove all of it. So the point being is that we got to start thinking about doing combinations of drugs. Here's another example. This is a patient with stage 4 intrahepatic cholangiocarcinoma. This is another rare tumor that occurs in the liver. It occurs within the bile ducts of the liver. We did mutation testing on this patient, and we gave them drugs that would target their mutations. And it turns out that response rates in this disease are somewhere about 15%, meaning if you give a drug, you're going to about 15% of the time you're going to see these tumors shrink. Now, in this scenario, in this patient's case, after a year of treatment, he'd almost had near complete resolution of his tumor with a completely out-of-the-box regimen, giving these 2 drugs, trametinib and nivolumab, which is a checkpoint inhibitor or an immunotherapy drug. If that's not impressive, what's more impressive probably is if you look at the tumor markers in his blood, they were actually over 10,000. The testing stops at 10,000. That's the upper limit of what it can show. And you can see here, after treatment, he'd basically gone down to normalized levels. If we look at that patient, that example of that gallbladder cancer, which is sort of in the family of these intrahepatic cholangiocarcinomas, but slightly different biology, you can see here this patient had pretty extensive disease marked by these green arrows. And we treated this patient with a combination of the 2 drugs that would create a 100% match. And lo and behold, we actually saw a pretty nice response in some of these tumors, and you can see, shrinkage of these tumors. Now, we're still learning though, and one of the problems that we recognized in this patient, and this was actually the first patient treated on the trial, was that we saw what we call a mixed response, meaning that these tumors that had been biopsied and we had done the sequencing on and we had the answer for, we saw a response in those tumors. However, you can see this one in red is something that wasn't there and grew, suggesting that there's more tumor heterogeneity that we're learning about. And so right now we're investigating how we can use circulating tumor DNA or liquid biopsies in order to better understand. So if we look at the entire patient cohort, the outcomes of these patients, we can look at those patients that got high matching versus low matching, and you can see here the response rates are much better. If we go back to that original description I told you of these ratios of the progression-free survival, they're a lot better in patients that got high matching. This translates into differences in survival, and this translates into differences in not only progression-free survival, meaning that the time it takes for the tumors to grow, but it also translated into a difference in overall survival. So we've learned lessons from this, and one of the things we've learned is it's important to do this as a team, and one of the parts of that team is having somebody to help us get the drugs and to interface with a combination of a couple things. One is the insurance companies, 2 is the patient advocate groups, and 3 is the drug companies in order to help us to get the drugs for these patients. Because often it's very easy for the insurance companies to say no because we're using some of these drugs off-label. The second thing we learned is that we need to start giving these drugs earlier on, meaning that we can't wait till these patients have failed 4, 6, 8 lines of drugs before we suddenly start saying we need to start doing this novel approach. We need to start doing it earlier. And so just to summarize some of the lessons we've learned, it turns out that these single agents are often not adequate to treat these types of cancers, and that we need to start thinking outside the box and start treating not only the one alteration that's identified, but targeting multiple alterations that are identified. And that we can start treating more of these patients in this manner. And that this really is a combination of using not only targeted agents, but also thinking about using immune therapies or immunotherapies, and really trying to personalize the therapy for these patients. And I'm just going to end on this just a quick last story here. This was one of our patients that was, story was told about her in National Geographic in January of this year, and our iPredict trial was highlighted here. While this is a common disease, breast cancer, this patient had triple-negative breast cancer, which is one of the rare, more aggressive forms of it. She had failed 6 lines of therapy, 10 different drugs. She had a bowel obstruction. I took her to the operating room. We got her sequencing done, and it turned out she had all these 13 mutations here. And you'd say to yourself, there's no way you can give somebody 13 drugs. You're going to kill them with toxicity. But she did have this, what we call high tumor mutation burden, meaning lots of mutations in her tumor. Well, it turns out that actually portends a potential role for immunotherapy or giving immune checkpoint drugs. And so I had her see Shumei Kato, who was one of my colleagues, and we started her on this regimen. And lo and behold, within the first 2 months, her tumor markers started dropping, and by about 6 months, she had had no evidence of disease. This was imaging at 14 months. She's actually about 2.5 years out now and has no evidence of disease after actually being referred to me with plans to go to hospice. So we're at the tip of the iceberg now with genomics that I'm talking about today. There's a lot for us to learn still about the RNA and about the metabolics in tumors, about using artificial intelligence. There's still a tremendous amount that we don't understand. And with that, I will say thank you. These are all the agencies that have funded my laboratory. And a particular thank you to our team, and most importantly, the patients and our families that have allowed us to treat them and investigate a better way to treat their tumors. And I'll stop there and happy to answer questions at the end. Thank you very much.
Okay, good afternoon, everyone. I'd like to thank Global Gene to invite me here. My name is Ye Liu. I work for a biotech company called REGENXBio at Rockville, Maryland. At REGENXBio, we use AAV platform to improve patient life. So today I want to give you a brief review of AAV gene therapy. Can I have a show of hands who heard of AAV? Okay, most of you heard of AAV, so I guess I can finish my talk now. So hopefully after my talk today, you can go home, know a little bit more about AAV gene therapy than you come here today. So what is gene therapy? According to the definition from FDA, gene therapy is a treatment approach that modifies a person's gene to treat disease. So normally, it works by 3 mechanisms. First one is most common one, is to replace the disease-causing gene with a healthy copy of the gene. Second one is inactivate the disease-causing gene that is not functional properly. For example, if a gene is overexpressed it too much, it becomes toxic, we can deactivate it, reduce the gene expression. And the third one is to introduce a new or modified gene into body. In this case, we may introduce an artificial or synthetic piece of gene into body. For example, in the case of CAR T therapy, chimeric antigen receptor, which is artificial synthetic gene. So most of the gene therapy work by these three mechanisms. So if we talk about there's also two types of approaches, right, for gene therapy. The first is what we call in vivo gene therapy. So in vivo means the gene modification happens inside the patient's body. In this case, a delivery vector, in this case AAV vector, carry a healthy copy of the gene, was made as a drug product, shipped to the patient, and directly injected into the patient's body. The gene modification happens inside of the patient's body. And the next case is what they call ex vivo gene therapy, means the gene modification happens outside the patient's body. Let this figure show that either the peripheral blood or bone marrow take off the patient. After cell culture, a vector was used, in this case a retrovirus vector, carry a piece of therapeutic gene, was used to transduce this cell. And the therapeutic gene got stably integrated into the cell. After further amplification, the cell was infused back to the patient. And so in this case, the gene modification happened outside of the human body. Sometimes this therapy also called cell therapy, such what you heard of CAR T, this type of therapy. So today, I'm going to focus about the in vivo gene therapy. So you guys heard of AAV, it's Adeno-Associated virus. It's been turned out to be a very powerful in vivo gene delivery vehicle. So gene therapy is all about deliver the gene to the human body, into the cell that's missing this gene. Scientists have been dreaming about gene therapy for several decades, but they don't have a good delivery vehicle. You can't just give a piece of DNA to the patient, it won't get into the cell it needs. So it's all about delivery vehicle. So AAV, this wild type AAV, as in this cartoon, the orange color is what we call the virus shell, or the virus capsid. And inside is a protein capsid. Inside is a piece of single-strand DNA, consists of two major viral genes. So AAV, what we need to know is that it's replication defective. It cannot amplify by itself. That's also the reason it doesn't cause any diseases. It's also it's what we call helper-dependent virus. It needs the help of another virus, such as adenovirus, to replicate itself. So in about 1980s, scientists from University of Florida found out that you can actually further engineer AAV. You can remove the whole viral gene, replace with a therapeutic gene. Then effectively, we turn this AAV into a gene delivery vehicle. Then that's why we call it a vector. A vector means a vehicle that delivers the gene into the human body. So a very safe feature of AAV is that it's not known to integrate into human genomic DNA. So we have a very low chance of integrating into where it shouldn't be, disrupt normal cell function. So this picture here is what you see if you observe AAV under a high-power electron microscopy. This is what you see. The blue color is a virus shell or viral capsid. It's the virus protein. And you see this orange color spikes or bumps outside of the virus shell. These are the variable loops on the virus. So these spikes determine how AAV can bind into the particular receptor on different cell type. There's many different AAV subtypes. They have different bumps, different spikes. So this feature allows them to enter different cells in human body. So like I said, there's many AAV subtypes, and originally people discover AAV2 to AAV6, and then the second generation have up to 100 different AAVs isolated from human and primate. So each AAV has its own unique specificity to a different organ. For example, such target brain better, some target liver better, and some go target skeleton muscle better. So based on the particular disease, we have the option to choose appropriate AAV to deliver this gene. So this two pie chart means the current AAV gene therapy in clinical trials. As we know, the AAV2 is the first one got discovered. So most of the gene therapy trials are using AAV2, but in recent years, the novel AAV vectors such as AAV8, 9, 10 are becoming more and more popular. And the primary target of AAV, based on its tissue specificity, are mostly brain, muscle, liver, and eye. So is there any AAV gene therapy being approved in the US? Yes, there is. The first one is called Luxturna. It's approved in December 2017. This drug uses AAV2 to carry a gene called RP65 for a type of blindness. The second drug called Zolgensma is approved this May. It uses a virus called AAV9 to carry a gene called SMN1 for spinal muscular atrophy. So these are the two drugs, AAV drugs approved in the US, but there's many, many more currently in clinical development, up to I think at least several dozen, still counting. There's new popping up every day. It's still a relatively new therapy, so most of the clinical development are in phase 1 or phase 2, but there's a few in the phase 3 late-stage development, such as hemophilia A and hemophilia B are good examples. So, like I said, AAV is what we use to deliver the DNA into the cell. But how do we deliver AAV to the human body? Now we are talking about the route administration of the AAV. The most common method is what we call systemic. Basically, an intravenous infusion into the vein. In this case, it directly goes into the blood circulation, so the AAV will mostly target liver. After liver got saturated, it will target the skeleton muscle. And next major organ is the central nervous system. Some type of AAV, such as 9 and 10, can target the neuron very good. So we can directly infuse AAV into the central nervous system. In this case, directly inject into the spinal column, what we call intracisterna or intraceca, or you can infuse into the ventricle of the brain, which we call the ICV, and also we can infuse into the brain matter, which we call intraparenchyma. And finally, the ocular administration. So eye is a very good organ for AAV gene therapy. First, because eye is small, so it doesn't need a lot of viral vector. And also, the eye is a relatively immune-privileged or immune-separated organ. So it doesn't get messed up by the body's immune system too much. So there's two main injection methods. One is called subretinal. It's delivered to the back of the eye. The approved drug Luxturna using this method. Another method called intravitreal, which is injection into the middle of the eye. So I want to touch about cross-protection. I think there's a question about this in one of the morning sessions. For certain type of disease that involve a secreted protein or enzyme, we actually do not need to correct the gene of every single cell. In this case, we deliver the AAV, and the AAV, in this case, in fact, will transduce a single cell, make the protein it needs, and then this protein got secreted outside of this transduced cell and got picked up by the neighboring cell. and correct the function of the neighboring cells. In this case, one transduced cell have the ability to correct many other cells. But this mechanism will only work for the enzyme or protein that can be secreted and picked up by the neighboring cell. Most of the lysosome storage disease use this mechanism and can be treated this way. So in this slide, I want to touch about the success and challenges in the in vivo AAV gene therapy. The more green color means more success. The more red color means more challenges. This is also a slide I call the 6 R's, because it started with the 6 letters start with R. The protein replacement therapy is what most of the current clinical trial and approved product are based on. This is something the field is getting pretty good at. The reduce means to reduce the overexpressed toxic protein. This again is something that scientists are working on but haven't entered clinical trial yet. Repurpose is what I mean when they deliver the gene of a therapeutic protein used as a gene therapy to repurpose a normally antibody into gene therapy. There's just a handful of clinical trial working on this, no approved product yet. And the final 3 red boxes are something the field is actively working on, but we haven't been doing very successfully yet, which is redosing, means managing immunogenicity, regenerate, which means treatment post-tissue damage, and regulate, which means that we can turn the AAV gene therapy on and off, because the current therapy is in the always on state. We cannot really turn it off. So what rare condition, I mean, each of you all concerned about your own rare condition, what condition are most likely to benefit from AAV gene therapy? The first, I would say, it has to be a monogenic disorder. If the genetic mechanism is very well understood, you know what gene is missing. If you supply back this gene, it's going to help. And the protein sequence should be generally less than 4 kilobase pair, because AAV have a limited packaging capacity, 4.7 kilobase pair, and you need to leave some space for the regulatory elements such as a promoter and enhancers. And we should have well-studied animal model, large mammal preferred, which will make the translation from preclinical to clinical much easier. The target organ are eye, liver, muscle, central nervous system at this point. And clinically, because you know rare disease, it's hard to do a large placebo-controlled trial. So if you have good nature history and good biomarker, it will make the AAV gene therapy clinical development easier. So how long will the benefit of AAV gene therapy last? This is the question many people ask, but no one have a very good answer yet. So I would say there's evidence for more than 10 years benefit in large animal studies, and also from early hemophilia patients. We are still counting. Like I said earlier, the safety feature of AAV is that it doesn't get integrated into the human genome. But at the same time, AAV can get diluted after the tissue growth and tissue regeneration. So that bring a good question of what is the appropriate age to receive a gene therapy. And I think that's very dependent on the particular diseases we are talking about. For example, for the Zolgensma to treat the spinal muscular atrophy, the FDA approved the drug to be given to the patient less than 2 years old, because in that particular disease, most patients don't survive past 2 years old. So it's very important to give them the treatment as early as possible. But however, for other diseases such as hemophilia, where there's a pretty efficacious standard of care, giving the enzyme replacement therapy or factor 8 to 9 therapy. In that case, we may want to wait until the patient grow a little bit bigger, more like adult or young adult, for us to give the AAV gene therapy, so their efficacy can last longer. Because of the AAV is still a virus, it doesn't cause disease, but the human body still sees as a foreign virus. So after we give the AAV gene therapy first time, human body will generate a large amount of neutralizing antibody to prevent the second dose. So right now, the second dose is still not possible, but scientists are actively working on multiple ways to solve this issue. So we talk about dose for AAV gene therapy. It's quite different than the small molecule dose you normally think of. Because AAV is very small. It's only 20 nanometer in diameter. Your hair is 5,000 times thicker than the AAV. So this is, because it's very, very small, for a normal ocular or eye indication, we need 1 times 10 to the 11 virus per eye, which is 100 billion virus to inject into the eye. For systemic indication, we use a number of viral genome per kilogram of weight. A normal dose is 100 trillion virus per kilogram of body weight. For a normal adult, we need 8 quadrillion virus, which sounds like a humongous amount of virus. It's such a large number, I couldn't even count the zeros. But if you actually calculate the weight of this 8 quadrillion virus, it's only 70 milligram. 70 milligram of a drug is a normal small molecule drug you take every day. So that's what we use the dose for AAV gene therapy. So the current advance in AAV gene therapy, I would say we are discovering novel engineered AAV capsids to improve precision of tissue targeting, so it can go to a particular tissue much better. And second, there's a regulatory element such as promoter, enhancer that can allow more robust gene expression, also allow tissue-specific gene expression. These are the 2 items that I currently work in my lab. We are working on improved capsid and the transgene. And another point I want to make is, AAV is a delivery vehicle. It can use to deliver a healthy copy of gene. It can also use to deliver CRISPR element. The combination of AAV and CRISPR can allow in vivo gene editing. There's better immune modulation to improve safety and prolonged efficacy, because like I said, AAV can have immune response in the body. So if you use certain immune suppression to modulate, it can increase the duration of benefit. We are making better assays for neutralizing antibody and biomarkers. I think they was touched this morning as well. Some of us gonna have neutralizing antibody for one or other type of AAV. So we need good assays to measure this to determine who are eligible for which type of AAV gene therapy trials. I mean, there's this AAV, we need trillions of virus to make it. It's how we're going to make it. So the pharmaceutical company are building large-scale manufacturing facilities, 500-liter, 2,000-liter bioreactor to make all these trillions of virus as drug. So the take-home message of today's talk is AAV may be a safe gene delivery vehicle. It's currently a one-time administration, but the efficacy will be long-term. We are talking about at least years of efficacy. The target working are mainly eye, liver, muscle, and central nervous system at this time. And the AAV gene therapy may require immunosuppressant, and the redosing is not possible at this time. I want to point it out that this is a very extremely active research field in the past few years, especially after the recent 2 approved drugs. And there's a lot of breakthrough happening every year in terms of basic research, translational research, clinical development, and also manufacturing science. So these are some of the websites that you can learn more about AAV gene therapy. You can take a picture to read more about those. I will point it out, the American Society of Gene and Cell Therapy have a very good clinical trial finder for you to find out all about the gene and the cell therapy clinical trials. NIH, NORD, and FDA have good websites discussing about gene therapy. World Symposia have a good collection of lysosome storage disorder if you're interested in that. And our host Global Gene actually have a good toolkit to talk about gene therapy as well. So I'm going to end here and take questions together with Jason.
Great. Thank you, Yi and Jason. Appreciate that. And just a reminder in the room, We have the mobile app. I've got a couple questions here, so thank you for folks who are viewing online and for in the room. You can send me, if you're too embarrassed or too not willing to get up here and ask the question live, you can send it to me on the phone app, or you can join me here at the mic. So I have a couple of questions, and Yi, I'm going to start with you. What is the future potential? What future potential is there for gene therapy for targeting the skin? Thinking about any potential for the DNA repair disorder, xeroderma pigmentosum.
To target the
Gene
skin?
therapy targeting the skin.
Targeting the skin.
Is there any potential?
I think that's a good question. I haven't read any report of using AAV to target skin. There's other virus has been using to target the disorder in skin, but I haven't seen report of AAV yet.
Okay, Jason, this next question is for you. What makes a patient evaluable?
Any patient's evaluable. It's a matter of basically getting like in the situation of a tumor, getting the tumor sequenced and potentially determining whether there's something where a drug can be used to target something within that tumor.
And from your case studies, was there something about those patients that made them more evaluable than others?
Not necessarily. We haven't published the data yet, but we're just finishing accrual right now of another arm of the trial where we're actually doing it in the first line, which was probably the most controversial part in terms of getting it through our institutional review board, is basically saying we're going to take patients and not give them the standard of care drugs, but we're going to give them molecularly matched therapies based upon those treatments. So I think it all has to be done under the setting of a clinical trial and not just sort of Wild West. But anybody potentially could have a tumor and/or other diseases. And I think that's probably going to be the next extrapolation. In talking with gastroenterologists at our institution, is can we start thinking about using this sort of paradigm for things like Crohn's disease, inflammatory bowel disease, ulcerative colitis, all these other things that are problems.
Okay. Any questions? Here we have a line here. Great. Thank you.
Thank you for your presentations. Very, very informative, Dr. Liu. And just want to understand a couple of things. Are you going to be putting your slides online so that we can go and read and learn? And the second thing is, so I'm funding some research on SRD5A3, which is more enzyme, and it's a nonsense mutation, small cell. And we're looking at partnership with pharmaceutical biotech companies. So does your company collaborate with foundations to do research or partner on the AAV, primarily on the retina or on the brain side?
Sure. Thank you very much for the question. And the first, I think the slide, if the organizer okay, I'm certainly happy to share the slide, and it can be shared through on the online stream as well. I can also send you the slide as well. So the second question, why don't we talk off the stage about the future collaboration, and we are certainly happy to collaborate with foundations.
Great. Thank you.
Thank you for your presentations. We were told by one of our researchers that gene therapy was only applicable to recessive diseases. The disease I have is autosomal dominant. Is that true, or looking for some clarification on that?
Sure, that's actually a very good question. And remember in the first slide, I talked about the three mechanisms of AAV gene therapy. The first one is gene replacement or protein replacement, which is more appropriate for the recessive disorder, if you're missing something. If you have a dominant disorder, which sometimes lacks, you're producing a protein that is toxic, right? It's inhibiting the other healthy copy of the gene. It's toxic. So in this case, we will need a inactivating mechanism. We will need to produce something that inactivates the toxic piece of the gene. So most likely, it's going to work through a mechanism called RNA interference. We'll use microRNA to inhibit this toxic protein or toxic RNA. So that's one mechanism. And another mechanism is if we have an antibody that can target this toxic protein, we can use this, also we call vectorized antibody or antibody gene therapy. We can use AAV to deliver this therapeutic antibody to bind to the toxic protein. So I'll say yes, the AAV gene therapy is capable of treatment of autosomal dominant disease, but I think it's a few years behind the protein replacement therapy.
Thank you very much.
Sure, pleasure.
Hi, thank you guys for being here. I have a rare disorder called the MYH9 related disorder, but I have 3 questions for you on your lecture. With the AAV, you said that you saw antibodies develop towards AAV. Do you also see an autoimmune disorder develop with those antibodies? And then what diseases are candidates for the AAV and CRISPR combination?
Thank you for the question. And I think for AAV, there's a neutralizing antibody, and there's also certain T cell response the human body can generate that limit the efficacy of AAV. But I'm not aware of any report talking about autoimmune against AAV yet. So second question of AAV and CRISPR gene editing. Right now, there's a clinical trial to use AAV deliver CRISPR gene editing for a form of inherited diseases in the eye, in a form of blindness. So that's currently ongoing because, like I said, eye is a relatively immune isolated organ, and so people worry about AAV gene editing in a whole way that how can we control the gene editing so that it only added the correct piece of gene, doesn't edit other gene. So I think there's a very active research field to identify the off-target efficacy. We want to reduce the off-target as much as possible. By doing that, there's just new paper published every month to talk about maybe we can limit the gene editing to just a small period of time. So we deliver the CRISPR, edit the gene, and then the CRISPR is gone. So if you can engineer in that sense, then it can reduce the chance of off-targeting effect.
Would that then also broaden the types of disorders that it could be used for if they were able to solve that problem?
Absolutely. I think that would be the hope.
Thank you.
Hi. Mike Raleigh, SNGAP Research Fund. Thank you both. Really great talks. Sorry, but it's another AAV question.
No, that's fine.
Our children have a neurological haploinsufficiency, right? And so we've been thinking a lot about AAVs for the brain. And having said that, 2 very broad questions. Everyone's always told me AAV9 was the only way to address the brain, and I saw AAV-RH10 up there. So could you please distinguish those a bit? And then specifically, is there a difference in the payload size? And then the other question, if you could get through the regulatory nightmare and deliver intrathecal AAV therapy for a given any neurohaploinsufficiency, what I've been told is you'd be lucky to hit 20% of the brain, but you can't titrate how much any one cell gets of the virus. So if you have a virus that is toxic over 100%, you could have a problem and kill off some neurons. And then my question as you look forward to future research is how much regulation and dispersion of the virus is currently being researched for problems like this?
Okay. Yes, thank you for the question. And you bring up a very good point. The first is about AAV9 and AAV-RH10. They are both used to treat neurological disorders, to target neuron. And they are both showing, efficacy. Like AAV9 is in the approved drug already. So it's efficacious in that particular disease, SMA. And RH10 is not used in any approved drug yet. There is, I haven't seen any side-by-side comparison in a particular disease model, so I can say there's not enough evidence to say who is better than the other. But I would say AAV9 is certainly being used in more clinical trials for neurological diseases. And the second question, and I would say there's a lot of research to study the promoter, the regulatory element, such as Dr. Stephen Gray talked about his GAN trial this morning, that I think based on the particular diseases and for example, some secreted protein, you may want to have stronger promoter. To want one cell to make more protein, it gets secreted to help the neighboring cell, what is called cross-protection. But for some non-secreted protein, that each cell is on its own, it cannot secrete. But you don't want this cell to express too much of the protein. So in this case, you may want a less potent promoter, but you want higher virus dose. You want to infect as much as cell possible, but in each cell, you don't want to express too many protein. So it's very disease specific, and a lot of the data we are generating in animal models to help to guide the dose for the human clinical trials.
Thank you.
We have about 15 minutes until the closing session. So you have a 15-minute break. We're going to go ahead and close this session, but there's a couple of folks who have questions here for you, so I'm going to ask you to step up to the side of the podium so they can answer your questions for you one-on-one after we close. But let's go ahead and thank Jason and Yi for their time this afternoon.